Homo Universalis video number 4, the Calvin cycle. Note, it is recommended that subtitles are turned on for this video. The simplified version of the Calvin cycle model begins with a molecule known as ribulose 1,5-bisphosphate, or RUBP. RUBP is a naturally occurring molecule found in all domains of living organisms. In a phase of the Calvin cycle known as carbon fixation, an enzyme known as ribulose 1,5-bisphosphate carboxylase oxygenase, more commonly referred to as Rubisco, conducts carbon fixation upon the RUBP molecule. Rubisco is the most commonly occurring enzyme on Earth. It works by utilizing carbon dioxide molecules to break RUBP apart into 3-phosphoglycerate. Some of the created 3-phosphoglycerate is diverted into other central metabolic pathways, whilst the other part stays within the cycle. From the previous light-dependent reactions which occurred within the thylakoids, we have many molecules of adenosine triphosphate, ATP, along with charged nicotinamide adenine dinucleotide phosphate H, NADPH atoms. These will now be used to break down the 3-phosphoglycerate molecules which we just derived. First, ATP breaks down the 3-phosphoglycerate into 1,3-bisphosphoglycerate, which is, on its own, a more stable molecule. NADPH uses its extra electron to destabilize this molecule and break it down into glyceraldehyde 3-phosphate, or G3P, and an inorganic, which means it does not contain any carbon atoms, phosphate group. The Calvin cycle then goes into its final phase, regeneration. During this phase, 1/6 of the made G3P molecules undergo several reactions to create sugar, like glucose and fructose. The rest 5/6 undergoes a plethora of reactions, which we will later talk about, to create ribulose 5-phosphate. Ribulose 5-phosphate is then modified by energy from ATP molecules into RUBP, which restarts the cycle. Now, let's go in depth into the regeneration phase of the Calvin cycle. After the reduction phase of the Calvin cycle is complete, we are left with 12 molecules of glyceraldehyde 3-phosphate, or G3P. In its pure form, this molecule does not offer any useful function to the plant, so it needs to be converted into another state. This is where the regeneration phase of the Calvin cycle comes in. Regeneration is the last of the 3 phases of the light-independent reactions and is one of the most important. Anyways, 10 out of 12 glyceraldehyde 3-phosphate, or G3P molecules will be used to continue the cycle, whilst only 2 will be used to make sugar. Before we go in depth into this phase, it is important to note that the conversion of G3P and other molecules, such as DHAP, into one type of molecule is done simultaneously along with their conversion into another type of molecule. Nevertheless, let us begin with what happens to the 10 molecules of G3P which will remain in the cycle. The process begins when some of the G3P molecules are converted into dihydroxyacetone phosphate, or DHAP, using an enzyme known as triose phosphate isomerase. Next, 2 enzymes, aldolase and fructose 1,6-bisphosphate, convert DHAP and G3P, excluding the 2 saved G3P molecules, by dihydroxyacetone phosphate into fructose 6-phosphate. Afterwards, another enzyme known as transcatalase removes 2 carbon atoms from fructose 6-phosphate, which creates erythrose 4-phosphate, or E4P. The 2 carbon atoms are then added to G3P to create xylulose 5-phosphate, or XU5P. Aldolase then converts E4P and DHAP into cetoheptulose 1,7-bisphosphate. Next, enzyme cetoheptulase 1,7-bisphosphate breaks down cetoheptulose 1,7-bisphosphate into cetoheptulase 7-bisphosphate. Trans-catalase then removes 2 carbons from S7P, which are then transferred to 1G3P, which produces ribose 5-phosphate R5P and 1 molecule of XU5P. Next, an enzyme known as phosphopentose isomerase converts the molecule of R5P into ribulose 5-phosphate or RU5P. Another enzyme, phosphopentose epimerase, converts the XU5P molecules into RU5P. Finally, an enzyme known as phosphoribonuclease phosphorylates or attaches a phosphate group to all RU5P molecules, which creates the long-awaited ribulose 1,5-bisphosphate or RUBP molecule, which allows the Calvin cycle to start all over again. However, what happened to the 2 molecules of G3P which have been saved? Those molecules are converted into either glucose 1-phosphate, glucose 6-phosphate, or fructose 6-phosphate. This is done through the following steps. First, the G3P is converted into 2 1,3-bisphosphoglycerate molecules using the ATP synthesized in the light reactions. Afterwards, NADPH uses its electrons to destabilize the 1,3-bisphosphoglycerate, which then breaks down into 2 glyceraldehyde 3-phosphate molecules, one of which, in a reversible reaction, is converted into dihydroxyacetone phosphate. The other molecule is made into fructose 1,6-bisphosphate, which then makes either glucose 1-phosphate, glucose 6-phosphate, or fructose 6-phosphate, all of which are sugars which can be used by the plant. This is a much more detailed model of the Calvin cycle, which is also sometimes referred to as the Calvin-Benson cycle. As we can see, the carbon fixation, reduction, and regeneration of ribose phases are shown in great detail.